What's a helpful way to think about and draw the structure of the nucleic acids, RNA and DNA? We start with a backbone, O, P, O, C, C, C. The OPO is a phosphate. I'm not showing the double bond oxygen, the OH down here. This would ordinarily be an OH, but it would be connected in a long chain to another carbon. The CCC represents part of the sugar, because the backbone is phosphate, sugar, phosphate, sugar, phosphate, sugar. Phosphate is OPO, sugar is CCC. What about the rest of the sugar? Carbon, oxygen, carbon, carbon. It's a pentose sugar, 1, 2, 3, 4, 5. There'd be an OH up here. If it's ribose, there's an OH here, but that would be missing if it was deoxyribose. This is a phosphodiester bond, so phosphodiester bond across here, and between the carbon and the next phosphate is another oxygen. We number the carbons. Remember that the carbon with 2 oxygens, or the closest carbon to the end with 2 oxygens, is numbered 1. In the case of ribose and deoxyribose, that carbon is the end one. There's the 2 oxygens. There's no other carbon off here, so that's 1, 2, 3, 4, and 5. So the phosphodiester bond goes between the 3 carbon on one end and the 5 carbon on the other. So the full backbone is OPO, CCC, OPO, CCC, OPO, with these diester bonds. You can also consider this between a sugar and something else, a glycosidic bond. Next, we add in the nitrogenous base, which I'll draw as a ring with a nitrogen, carbon, carbon, nitrogen, carbon, carbon. There's a hydrogen, the third bond on that nitrogen. Takes out an H2O and leaves behind this single ring attached to the 1 carbon of the sugar. We could also use a double ring for a nitrogenous base with 2 carbons joining them. Again, a nitrogen here, carbon, nitrogen. There's only 5 atoms here. Carbon, nitrogen, carbon, nitrogen. Again, there's a hydrogen here, an OH. We join them. I haven't shown you the amino groups, which most of the nitrogenous bases, whether they're a single ring or a double ring, have an amino group on them. That's part of what makes them a base. You can add an H plus to get an NH3 plus here. The phosphate, sugar, and nitrogenous base is a nucleotide. A chain, then, is phosphate, sugar, phosphate, sugar, phosphate, sugar, all joined by these oxygen glycosidic bonds. Each nitrogenous base is either a single ring or a double ring. The double rings are purines, the single rings are pyrimidines. Double-stranded DNA, we have phosphate, sugar, phosphate, sugar, phosphate, sugar as a backbone with 5 prime, 3 prime, 5, 3 carbon, 5, 3 carbon. And going the other way, we have a sugar, phosphate, sugar, phosphate, sugar. And in this case, we're going 5 prime, 3 prime, 5 prime, 3, 5, 3. Notice here we start with a phosphate 5, 3, phosphate 5, 3, phosphate 5, 3, opposite direction, phosphate 5,3, phosphate 5,3, and so on. Each sugar has a nitrogenous base, either a single ring or a double ring, or single ring. And there's hydrogen bonding between them, either 3 sets or 2 sets. So the strands are going in opposite direction from phosphate to sugar, phosphate to sugar. If they don't go in opposite directions, then the bond angles holding the double rings and the single rings don't match them up properly, and you don't get hydrogen bonding. We can draw this much more simply as A's, T's, C's, G's, A's, T's, T, A, with a line, and if we want to show hydrogen bonding, we can show hydrogen bonding, not worrying about the exact number. RNA has the same structure: phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Again, 5 carbon 3, 5 carbon 3, 5, 3, 5, 3. If this was the end of the strand, there'd be an OH group on that 3 carbon. At this end, there's a phosphate on the 5 carbon. Again, either a single ring or a double ring. RNA forms double strands, but it also easily separates as double strands. And RNA is different from DNA because the sugar, ribose, has an extra OH. That OH affects structure in 2 ways. One is it keeps the sugar from shifting back and forth between 2 conformations, which makes the whole chain a little bit more rigid and makes it harder to twist into the shape in order to form a double strand. It'll do it, but it also easily pops free. The second effect of that extra OH, that OH can interact with that bond and break it. So even though RNA is reasonably stable over a shorter time period, over months and years, it's a much less stable molecule. It breaks down more easily, and that could lead to genetic damage. So for 2 reasons, those OHs make it harder to stay in a double-stranded conformation and make it more likely to break. RNA is useful as a way to transfer information or for short-term structural uses, but it's not useful as genetic material.